first slide. So my name is Amit Pathak. I'm a professor at WashU. My lab does cellular mechanobiology. So we look at how cells sense and respond to extracellular matrix forces and so on. And we generally work in the area of cell migration, collective cell migration, and upstream of migration, there has to be force generation. So we are interested in a lot of questions dealing with how cells interact with each other and different types of matrices. So what my lab does is something I won't get into today because the topic is animal cell adhesion. If you're interested about my lab's work, then you can email me or look at the website. Today, I'll just focus on the topic, the animal cell adhesion, and the little subtitle is a survey because I want to give you an idea of uh, what cell adhesion is and why it is important. Actually, what it is the main focus, not why it is important. Um, if you have never, studied anything about focal adhesion, that's my assumption. So let's just get started. Please interrupt me anytime. One slide. I have about less than 30 slides, so it's not too much, and I can stop at any point. The goal is to understand whatever I end up presenting, not everything that I have to present. So, what is a focal adhesion? It is uh, it is really an adhesion with the environment. I'm focusing more on the environment, not on adhesion of cells with each other. So these are molecular complexes, and complexes are composed of a lot of proteins and components. So in this schematic on the top left first, so this is a cartoon of a cell. I should try to get my laser pointer safe. See if that you can see it. Okay. So these blue cylinder type things, these are integrins, and there are these complexes of focal adhesion proteins. There are a bunch of those proteins attached to these integrins. Inside, there is F-actin, those are stress fibers, and microtubules also are present, and they provide structural properties to the cell. But the adhesion is this interplay of integrins and focal adhesion proteins, and we'll get into the machinery of this in a little bit. And myosin 2 plays a role because that generates forces which are important in the assembly of focal adhesions. And extracellular perturbations, meaning matrix stiffness or stretching, any kind of shear, those all will become important in the assembly of adhesions. This is just the overall picture of the context in which adhesions kind of reside. So taking a step back, why are adhesions important? They play a key role in the whole process of mechanotransduction. And you're doing this boot camp for cellular mechanobiology, so mechanotransduction is, of course, very important. On a basic level, transduction is taking one type of signal and converting into another. So there is conversion of mechanical cues, which can be, statics, kinematics, material constitutive behavior, all of those things, and then convert into biochemical responses. If mechanical cue just ends up being another mechanical output, then it's not really mechanotransduction. It is a mechanical cue going into a biochemical response, or biochemical meaning different types of signals within the cell, gene expression, protein production, and so on. And where do these mechanical cues come from in mechanobiology? Cells lining the blood vessels and so on. Blood pressure can act as a mechanical input in cancer progression. Tumor stiffness changes. It is known that's how tumors are kind of diagnosed in a very basic level by feeling the tissue. In fibrosis also, there is a change in mechanics of the tissue. Needless to say, in mechanobiology, the reason we are interested in the topic is because there are lots of mechanical changes in the body. So those are mechanical inputs. And what we are interested in are mechanical outputs through this mechanotransduction. And there are three key steps, there are lots of steps, but three key ones. Mechanosensing, whatever the input is, the cell needs to sense it. Then transmission, meaning whatever forces were felt by the cells need to be transmitted across different components in the cell. Otherwise, it doesn't really does it doesn't really do its job. And then mechanosensing, if all of the forces were or the mechanical properties were sensed and transmitted, but the cell doesn't respond in any way, then it's all pointless, right? That is the whole feedback loop. And the reason we are interested in mechanosensing is, I'll tell you in a little bit, is because this is the more complicated of the three things. And it's composed of cellular contractility. Cell senses the environment and then responds by generating forces. And cytoskeletal proteins like actin and microtubules and so on, they do help in these mechanical responses. And focal adhesions are at the, at the very initial point of sensing, because if you want to sense something, you need to attach to it, or at least have a transient adhesion to that external environment. So focal adhesion establishes that mechanical connection. And going over those three parts, mechanical transmission is the transmission of information within the cell. In this experiment, I provide the citation here. A small bead, magnetic bead, is attached onto the cell surface, and a force is applied. What you see in these color maps are calcium waves. They are a quick response of mechanical input. And as you can see, the calcium waves kind of respond within seconds. And there's a peak on this plot on the right, and then peak subsides and then goes to a plateau again in about a minute or so. So mechanical response then, or transmission of mechanical information within the cell, is quite fast. So for that reason, we won't spend too much time on it. It's really fast and it happens, and that's the end of it. That's how basically the transmission of information throughout this cell is very fast. Mechanosensing is a little more complicated. It is happening over seconds and minutes. So what is happening here? So initially, there are transmitted forces that change the macromolecules around the focal adhesion complex. And forces unfold these initial proteins. Talin is one. So because of the force, the protein is going to unfold, sorry. It's like, having a kind of a spring. And when you stretch the spring, the inner loops of the springs will open up. And in those inner loops are these scripting binding sites for other proteins to come in and cluster. And this talin one is important because it connects from the integrin to the intracellular cytoskeleton, like focal adhesion fibers. And the forces unfold this talin, reveal these sites for vinculin and other proteins, and those proteins then come in and cluster and stabilize the bond. We'll talk a little more about it, but that's the basic process. And that's why forces are important. If forces don't happen, then these proteins will not open up, and they will not allow other focal adhesion proteins to come and cluster onto this initial binding and stabilize the bond. That is what is needed here. So lots of little bullet points here. I put them there, but we'll go top to bottom. So like I said, clustering of focal adhesion proteins is important for stabilization. And once a bond is stabilized, then it is bigger, basically, and it can now support more forces. So initially, if the bond is, let's say, one bond, then it cannot support a lot of force. If you increase the force, it will just rupture. Now, because of the force, the bond grew in size. Now it's some unit of 20. Now it can support more force. So if a cell were to increase its force, the bond can now support it. So it's more of a feedback loop then. And more proteins unfold, then there's the recruitment of other focal adhesion proteins, and the clustering happens. And that's the way, how mechanoresponse, which is the generation of force, feeds back into mechanosensing, which is the generation of, which is the sensing of the force. And mechanical signals then lead to other mechanosensitive signaling pathways. You might have heard about Rho and ROC and RAC, those kind of responses within the cell. And then let's say actin myosin proteins are generating forces, but they have to be produced by upstream transcriptional networks. Those also come into play in mechanotransduction. So overall, looking at the time scales, signaling pathways can activate over minutes. So in the front of the cell, if there is actin polymerization, it can happen in minutes. But the gene expressions that need to be upstream, they might happen over hours and maybe even weeks. And that's where the category of transcriptional and epigenetic process lies, and that also happen over hours and days. So these are like three different time scales at which things are happening. Initially, the sensing or initial adhesion can happen in seconds and minutes. The transmission of that information throughout the cell can happen very quickly within a few minutes. And then downstream protein production and protein kinetics, that might take several hours and or even days. So that's the context in which focal adhesion kind of operates. Now let's dig deeper into what the focal adhesion is and what are its different types and how does it grow. So very first thing is cell forms a nascent adhesion and very small one. And those are called focal complexes, and they will just disappear if they don't grow. So initially there's an factin fiber here, and these are integrins. These are the these are the receptors that will bind to the ECM. So if there is a nascent adhesion formed, and if focal adhesion is not, proteins don't come and stabilize this bond, they will just disappear in seconds. And for F-actin filaments are very important because they polymerize at the front of the cell and they continuously flow. So if they keep flowing, what will happen is these nascent adhesions will keep dissociating and reforming, and sometimes at the same site even. So that's called focal adhesion turnover. And if the velocity of these actin filaments is very fast, then this turnover is also going to be very fast. That means focal adhesion will not get a chance to really grow. They will just keep turning over. And so that's what I'm trying to say here. Faster actin flow leads to more unstable focal adhesions. And this operates like a clutch, meaning, if the flow of the actin is really fast, then it won't stabilize. If it's really slow, then there is more time for the bond to stabilize. And so this complex kinetics of focal adhesion leads to two types of bonds. I realize this is a very busy slide, and do not learn about slide making from this slide. I only made it so that even if you read it, it makes sense. Otherwise, I would not put so many words on one slide. So anyway, now let's focus on the box here. So focal adhesion kinetics then leads to two different types of bonds. There is a slip bond kinetics. So here what is happening is forces can rupture adhesions. If it's only a slip bond, the role of focal adhesions is, role of forces is to rupture any adhesions that are there. Now, because of the rupture, now what is happening is there will be new sites available for focal adhesions, right? So let's say on the cartoon here, if a force is arriving and it ruptures one of these adhesions. As a consequence of that, now there is a site available for the formation of new adhesion. So existence of forces really makes available more sites for focal adhesion. That is a way to form adhesions, really. So it is kind of counterintuitive, but that's the mechanism for slip bonds. Now, in contrast, catch bonds are more like the ones that I was talking about earlier. So what do forces do there? If there is a small bond that is available, the force will come and stretch on the bond. And that bond will now open up different sites for focal adhesion proteins to come and cluster. And those proteins will come and cluster and stabilize the bond. So the existence of force stabilizes the bond against the rupture. So think about these two again, I would say. So forces are at play in both of these types of bonds, but different mechanisms. In the first one, forces come and rupture the bonds so that new sites are available for new bonds, new focal adhesions. So that is the way forces are kind of enabling bond formation, but at a rapid rate. But on the second one, the catch bond, forces are just stabilizing the same bond. They're not allowing new bonds to, they're not really playing a role in new bond formation, but they are playing an important role in stabilizing any existing bonds. So, and these two like broad category of bonds are bond dynamics are at the heart of focal adhesion formation. And they operate in different situations differently, soft matrices versus stiff environments, or within a cell, front versus back might have different, might be, they might be amenable to different, one of these two different dynamics. Any questions on this slide? I understand this is kind of complicated. To try to explain it, but I'm sure a lot is unexplained so far.
I'm having trouble understanding the sentence. Actin filaments continually polymerize at the front of the cell and then flow backwards over the FAs. What does flowing backwards over the FAs mean in this?
Okay, that's a good question. So here, so actin is protruding onto the membrane and it's polymerizing at the front, but the depolymerization actin or polymerized actin is flowing backwards. So all of that actin is flowing backwards, and that is what is causing the motion, sliding motion onto the focal adhesions. So if that sliding is really fast, if the depolymerization is happening really fast, then there is more of a shear on focal adhesions, and that will disallow any focal adhesion stabilization. And on the other hand, if it is very stable F actin, then the velocity is going to be low. That means the bond will have more time to stabilize. So if the filament velocity is fast of F actin, that will lead to smaller lifetime of focal adhesions.
Is this how like a wave gets transmitted across the cell in the focal adhesions or something like that?
A wave of, you mean filaments, or wave of like ions?
Of like adhesions, I
Okay,
guess.
yeah. Yeah, that actually is, yeah, that might be the case. It's not, so there are slipping bonds and sliding bonds and catch bonds and so on. The truth is somewhere in the middle there. Maybe the cell moves before the focal adhesions move. But if you see a sliding focal adhesion going one place to another in a cell, this might be the mechanism. Any other questions here?
Dr. Pathak, I
Yeah.
had a question. A very cool talk so far. I was wondering if the presence of focal adhesion depends on cell binding sites outside of the cell, and if a cell could mix up, say, like I asked this question yesterday to another presenter, mix up perceived stiffness with increased, like say, collagen ligand density outside of the cell.
Yes, that is definitely possible, and it does happen. So there is a study. There are papers showing that if ligand density changes, focal adhesion area, focal adhesion elongation, all of those things change. I'll present some of that later, and then we can revisit that question. But that's definitely possible. Even the dimensionality of those ligands is important in 3D versus 2D. There might be different mechanics going on. Even it doesn't need to be like a 3D collagen network, even a flat substrate. But if it's wrapped around like in a microchannel, then the presentation of ligands will have an effect on how focal adhesions evolve. So all of those things do play a role.
I see. Thank you so much.
Okay, moving forward. So looking more over this focal adhesion clutch on the left is soft ECM, or those two things are analogous, meaning if it's a soft ECM versus a slow or a low amount of force. So both of those things will have the same effect. So in this, on the left-hand side, what's happening is exchange rates, meaning bond formation, and dissociation rates are very similar. So bonds are continuously being formed and reformed. So a lot of turnover going on. So as a result, the lifetime of a given focal adhesion is going to be low. It's not going to be high, at least. So none of the focal adhesions have a chance to really grow. On the right-hand side, if it's a stiff ECM, or high forces are, high level of forces are there. So on the left-hand side, then both types of bonds can operate. Slip bonds, what will happen is there won't be any strengthening like before, and the exchange rates are going to be still high. And that will lead to smaller, again, lifetime of focal adhesions. On the right-hand side, on the catch bonds, because of the stiff ECM or high force, now the focal adhesions have higher chance to stabilize. More focal adhesion clustering will happen, and those same focal adhesions will last longer. Okay, so these two, so that's how slip bonds and catch bonds operate across different stiffnesses, and that gives you a mechanism as to how a cell might sense a soft or a stiff ECM. Moving forward, now this is if a cell is migrating on a given ECM, it's not a soft or a stiff ECM, just any given surface. Now within the cell, there are gradients of forces available. So in the front of the cell, these are protrusions. So like I was talking about earlier, there's a lot of actin filament formation and depolymerization going on at the front. So very high velocities basically of actin. If there were, and those have been kind of, if I were to give a talk on protrusions, I would present that data showing the actin flow rates being very high in the front and not that high in the rear. So that what that leads to is, based on the slide before, is more focal adhesion complexes, meaning focal adhesions of small lifetimes at the front. But that doesn't mean they don't exist. They keep turning over. There's a lot of those, many of those, but of smaller lifetimes. And in the rear of the cell, now actin velocities are lower. So any adhesion that is formed has a lot more time to stabilize. So that's what leads to higher and bigger focal adhesions in the back and smaller lesser, not very long-lived focal adhesions in the front. So you don't need really a soft or a stiff ECM to observe these kind of different dynamics of focal adhesions. They can exist within a given cell. And as a result of this gradient, now focal adhesions in the back are stronger, focal adhesions in the front are weaker. That gives you a polarity within a cell. Now cell knows its front and back, and that gradient or difference in forces and difference in strength of focal adhesions leads to migration. If the front focal adhesions were exactly the same strength as the back focal adhesion, there won't be any migration. Cell will just pull on both ends and will not have a net force and will not migrate. So this slip versus catch bond dynamics existing within a cell is an important cause of migration of adherent cells, at least. Okay. Moving forward, looking at these different steps of adhesion maturation, this will be important. This is also important to understand. On the left-hand side, the very first step is the integrin binding to the ECM, and then tailin protein coming into picture, and that makes a connection with the with actin filaments right next to it. If that doesn't happen, then the adhesion is not really communicating with the forces within the cell. Our 2,3 complex is basically just actin bundling and branching. It's not very important here, at least. Now, these focal adhesion complexes, then they can only develop or mature if there is clustering of other proteins. So vinculin and others, alpha-actinin will come and allow actin bundling. And over time, these focal complexes will come together and make bigger adhesions, so clustered adhesions. So that is the idea of going from nascent adhesions to focal complexes to adhesions. That's how they will grow. This we already talked about. It was kind of repetitive. So here, this is a summary of how the gradient of these processes within a cell. On the front, actin and focal adhesion kinase, paxillin, these are important. This feedback loop through RAC is important for turnover of adhesions that leads to shorter lifetimes. The Rho activation is kind of low in the front because Rho and RAC have this antagonistic relationship. And in the back of the cell, RAC activity is low, meaning actin dynamics is low. So actin filament velocity on top of adhesions is low. That leads to higher lifetime focal adhesions. And the focal complexes have a higher chance of becoming adhesions in the back of the cell, and similar to in the behind the lamellipodium. So this is something we talked about. These are the important kind of key candidates, signaling candidates in these processes, and they connect focal adhesion maturation with cytoskeletal forces. And you can look over these carefully and ask me if you have any questions about this. Some of this, in terms of focal adhesion dynamics, we already discussed, but this slide more talks about how it all connects to force generation and migration within a given cell. Okay. Another summary slide, I think I shared this article with you, is this is how this kind of illustrates a feedback loop here. So adhesions are made with the ECM, actin fibers connect with the adhesions, they pull on the adhesions, then adhesions are maturated. That adhesion maturation leads to a proactive signaling of Rho GTPases, and that leads to higher forces. That's the feedback loop that I was talking about earlier. So integrin signaling is, so it's not just a one-way thing that if you apply a force on focal adhesions, they grow. That's not it. That leads to downstream signaling of activation of Rho A, let's say, and that leads to further activation of forces. So this feedback loop operates until there is a stability within a given cell. Okay. What else happens in force generation is if there is a force being applied on the membrane, like I was saying, because of this feedback loop, force will grow. On the membrane, there are these ion channels. And if the force is applied, the ion channels will open up. Some of this, I think, I was gathering from the discussion from previous talk. We talked about this ion channel transport, and these, let's say, calcium ions. Then the transport will go up, and those calcium ions themselves can lead to higher force generation in the active myosin motors, and that again reinforces the feedback loop. But the feedback loop will not just keep going on forever. If the extracellular matrix is soft, let's say, it will not support forces, then the feedback loop will stop, meaning that it will stall, and the force generation machinery will only work until the force is achieved that the extracellular matrix can support. If the extracellular matrix is really strong, meaning like a rigid plastic or glass, then it will keep going until the focal adhesions can grow to the maximum size. And at that point, cell cannot even migrate, like the situation I was telling you earlier, the front and the back, they're all the same. There's no gradient of focal adhesions within the cell. Then focal adhesion size is maximum, but the cell is kind of stuck. So that is the other extreme of feedback loop going completely haywire. Okay. Another possibility is because of this, forces can be generated by the cell and the ECM. So let's examine this cartoon a little bit. On top, let's say this red dot is the reference point on the ECM, and green is a focal adhesion with a low amount of force, and these blue lines are the actin. Now there are multiple possibilities in different ECMs and in a migrating cell. Let's go from left to right. On the left-hand side, there is a soft ECM, and that is being pulled. Let's say soft ECM is being pulled, but because it's soft, it's not going to be able to support a lot of force. Then the focal adhesion force is yellow, meaning not so much, moderate force. But on a stiff ECM, now cell is generating more force. So the orange arrow is now on top of the cell. And because of the force generation, now focal adhesion will be under a lot of force. So it will grow bigger. Now on the stiff, let's say the soft and stiff is gone on a given ECM. If this matrix is being pulled like a extracellular pressure or something, then also focal adhesion will experience a lot more force and it will grow in size. And in a migrating cell, now this is both balanced. The force generated by the cell and the force supported by the ECM, they are in balance. Then the focal adhesion size is going to be moderate. It's not going to be either low or high. So these are different possibilities for focal adhesions, forces to exist and how they will grow or dissociate. Right. Going forward, now changing gears a little bit, what are focal adhesions made of? Before this slide, what we talked about was what kind of different bond dynamics focal adhesions can experience and what kind of different scenarios that can generate for the cell and the focal adhesions. So, and that big, oval, I was talking about focal adhesion proteins, it's kind of a black box so far. let's now examine quickly what kind of different focal adhesion proteins exist and what are their different roles. And like this slide says, check out this paper. Also, there are hundreds of proteins that can play a role in focal adhesions. Exactly which proteins do what is not yet clear. There's still papers coming out, until you can find some this week even, what are different roles of these focal adhesions proteins. But there are three main classes, and this summary I kind of agree with. So these are, some are ECM binding proteins. They're closest to the ECM. So integrins, different types of integrins, different subtypes of integrins bind to different proteins. This alpha B beta 3, this is a fibronectin binding one. For collagen 1, there will be different collagen 4, different types of collagen, there will be different receptors. So those are, that's one, those are ECM binding focal adhesion proteins. Second is actin binding. So if there are no proteins that attach this focal adhesion to the interior of the cell, then all of this will fall apart. So these are F-actin binding proteins, the main ones being alpha-actinin, vinculin, and talin. Like I was saying earlier, when these proteins open up, they expose binding sites for other proteins to come in. And those other are this magenta square here, core proteins. And that's where a lot of the ambiguity is, which one does what, but mainly paxillin, zixin, focal adhesion kinase, they come they form the core protein complex, and they are the ones important in stabilizing the focal adhesion. So in a sense, if you have ECM binding proteins, then the nascent adhesion will form. But if you have nothing else, it will quickly dissociate. Now, that nascent adhesion needs to connect to the cell to make a focal complex. Then come in the actin binding proteins, meaning the alpha-actinin, vinculin, talin. Now it becomes a focal complex. Now you have a starting of a bond. Now this bond needs, even if you don't have the core proteins, this bond will also dissociate in seconds, and new ones might form if the conditions allow it. If the core proteins come in, these paxillin, zixin, focal adhesion kinase, they are needed to come and stabilize the bond, and then it becomes a focal adhesion, and that bond now can grow up to a couple of microns in size. In the bottom of the slide, it says the average speed of these different types of molecules. There is this speckle microscopy to look at how different proteins kind of move as the cell is moving. Integrins, their velocity is kind of low. They are closest to the ECM, so they're not that likely to move. They're attached. Focal adhesion, these core proteins are next ones, and these actin binding proteins are fastest. So essentially, as you go further away from the ECM, the speed of these proteins, these molecules, goes up. So that's what, and that kind of speaks to their stability as well. The actin binding proteins are perhaps being turned over more, and they're coming and going, but still stabilizing the bond. And here on the right-hand side shows the velocity vector correlation. How coordinated are these molecules with each other? Meaning these actin binding proteins, even though their velocities are high, they're very coordinated. So that means they're kind of keeping their spatial localization much more so than core proteins. Core proteins are coming and going. They're not as well correlated as with each other as our actin binding proteins. So this is, I guess, one, I think this is the only slide I have showing the different components of focal adhesions and what are their different roles. So if you have any, this is an important one, mainly because if you do experiments down the road and focal adhesions come into picture and somebody says, what is the role of focal adhesions in this phenotype? Let's say you're studying cell migration or process like epithelial mesenchymal transition or any number of things. In mechanobiology, adhesions are important. And then somebody says, go look at the role of focal adhesions. Now that presents a problem, that which of these proteins would you be staining for? And there are lots of possibilities. As you read papers, focal adhesions sometimes are just defined by vinculin concentration or paxillin or talin or focal adhesion kinase. And how those choices are made, the rationale should be based on what you're interested in. If the rationale is looking at a subcellular process, let's say the role of force generation, then you should look at perhaps a focal adhesion protein that binds to F-actin. Then you should look at vinculin or talin or alpha-actinin. On the other hand, if the key premise of the study is changes in the ECM and how they affect adhesion, then it is probably better to look at paxillin or zixin, because those are the core proteins that are connecting the cell to the ECM. On the other hand, a question was posed earlier, if it is more about the ECM presentation itself and the type of ECM, then you should be looking at ECM binding proteins to examine the role of adhesions. Then you should look at the right integrin subtype, or if other receptors are involved, there are DDR receptors and so on. Then you should be focusing your study on those kind of focal adhesion proteins. So being aware of these different subtypes is, I think, very important and will be informative for your experiment design. Any questions on this or things so far? Okay, I'll move on, and welcome to come back to this if something comes up. So now some observational things, the properties of focal adhesions. In this experiment here, these micro patterns are allowing the cell to spread to a certain size. So if the cell is allowed to spread to a bigger size, the focal adhesion assembly kind of goes up as well. So the size of focal adhesions goes up. They're distributed at the edges, but the size goes up. In the bottom one, what it's saying that if you have the same ECM content, so if you look at these gray circles on the left-hand side in panel A, so this same amount of protein, if it is distributed over a bigger size, then what happens? So the result is on the right here. Now this is one cell sitting on this area. Now the same ECM protein on the bottom one is distributed over a larger area. Then the cell spreading goes up and the focal adhesion size also goes up. So essentially focal adhesions and the spreading size go hand in hand. So if the cell is spread to a larger area, the focal adhesion size also goes up. So even if you allow the same amount of ECM, but it is concentrated in a small size, the focal adhesions will not form that much at all, really. So they need room to spread. And the reason they need room is when the cell gets bigger, it is getting bigger by generating higher forces. And when the forces are higher, that means focal adhesions will become more stable and they go bigger in size. And that's the basic feedback loop we were talking about earlier. Similar observations here. If now in terms of stiffness, earlier I was telling you the conceptual framework. Now this is the evidence here. This is on glass and different ECMs. On top are actin fibers. Those are the force generating elements within the cell. So glass, 150 kPa, 4 kPa. The cell shape is very different. And if you look at the focal adhesion distribution, which is in green and yellow, their orientations are along the direction of actin fibers, meaning whichever direction the focal adhesions is being pulled in, that's the direction it's going to grow in. And they become bigger and bigger with higher, more actin fibers and higher forces, which itself depends on ECM stiffness. And initially, even if they look similar at 1 hour time point, over 6 hours, they become different. 2 megapascal versus 5 kilopascals. Their orientation and their shape and their size is different. And this is an important experiment as well, showing what happens first. So it's a chicken and an egg problem. Do you need forces to stabilize adhesions, or do you need focal adhesions to generate forces that will then come and stabilize the adhesions, right? So how do you decouple it? And the one way to decouple it by just plotting both of those things over time. So here, this is over from 24 minutes over time up to 300 minutes. What they plotted was focal adhesion orientation and the cell aspect ratio, meaning the idea that is the cell elongating first, then focal adhesions are responding to that elongation, or focal adhesions are elongating or orienting themselves first, and then cell is responding to those focal adhesions. So this kind of decoupling of those two concepts kind of clarifies that feedback loop. So on the top panel, top right, here is the focal adhesion orientation. So within an hour or so, focal adhesions start to elongate. That's what it is showing. And they keep elongating by about 4 hours or so. You know, they seem pretty elongated, these focal adhesions. However, if you look at the cell aspect ratio, up to about 2 hours, cell is quite round. Lower aspect ratio meaning a round cell, higher aspect ratio meaning an elongated cell. In about 3 hours or so, even the cell hasn't responded yet. But if you look at focal adhesions, they are already quite elongated. So what this plotting, simple plotting of data shows is that focal adhesion elongation happens first, and the overall cell kind of responds to it. So it is an inside out kind of a response or bottom up response as opposed to a top down response. So focal cell, overall cell shape responds to focal adhesions rather than focal adhesions responding to the cell shape. And so this is an important result as well. All right, this is falls more in the obvious category, is that if the cell is responding to elongation, sorry, if the focal adhesions are causing cell elongation, the nucleus responds as well. Now, in this case, this is a nano patterned surface where focal adhesion elongation is kind of locked in place. Focal adhesions will not grow laterally, they'll only grow parallel to these lines. And as a consequence of that, cell also elongates along these lines, and the nucleus kind of responds to it. And so this is more of a validation of that idea. If you only allow focal adhesions to grow in one direction, the cell is kind of forced to grow in that direction as well. And the nucleus will just go with it because the forces are compressing the nucleus in that direction. After this slide, I'll explain some models. So if you have any questions about the basic idea of focal adhesions, I'm happy to answer now. Wait, I think. All right, I'll talk about this one and then get into the model. So another interesting result is that if focal adhesions are growing in size, they're supporting more force. But is that linear or not? Are they supporting the same amount of stress? So this work kind of shows that. So on the left here, this is a focal adhesion area, and on the y-axis and the x-axis, focal adhesion force. So essentially, it is saying is that bigger the focal adhesion, higher the force. So it's supporting is the outcome. It is validating what we have been talking about earlier. And they've found out that if you divide the area by the force by the area, you get the stress, and that constant stress is about 5.5 nanonewton per micron square. So it's a very cool result. One caveat here is that this is only valid for focal adhesions, not really focal complexes. So more so for catch bonds, not so much for slip bonds. So any focal adhesions that are smaller than 1 micron square, as if you can see here, the area of focal adhesion kind of starts around 2. And the note in the paper is that the focal adhesion size is smaller than 1 micron square. Then the force they support is all over the place. It doesn't go linearly, because those are then very transient processes. A very small adhesion can support a large amount of force for a few seconds, then it will rupture. So then this linear relationship doesn't really hold. This is only true for more stable and bigger focal adhesions, not so much true for transient adhesions that might be present in the front of the cell. Okay. This is I think one last thing I'm going to talk about in terms of focal adhesion force relationship. In this experiment, these are microposts from Christian's lab, and the cell is sitting on it. Those you might have seen. What is different about this one is there are these nanowires, magnetic nanowires, inserted randomly into some posts. And what that does is that then you can actuate this post. So you apply a magnetic field, this post will get actuated. So you're basically applying a force uh, at a at a given focal adhesion to see if that increases the size of focal adhesion or not. So earlier I was talking about if the forces, focal adhesions grow wherever the force is higher. Here you're applying the force and see if focal adhesion grows or not. So here in a single actuation in panel C, focal adhesion grows. And if you do multiple actuations, focal adhesion further grows. So this is again validating the idea. If you have started making a focal adhesion and you come in and externally apply force, that focal adhesion will then grow. It doesn't need to be cell generated force. It can be an externally applied force. So that also further kind of decouples that chicken and egg problem. What happens first, force or the focal adhesion? It is force. Now I'll talk about, and these are all different experiments, different uh, theories behind how focal adhesions operate. That leads to different types of models, computational and mathematical models to capture those processes. And when it comes to models, the simpler the better. But then the problem is if it's really too simple, it won't capture everything about a biological process. But let's start with with some simple models. So I'll give you 3 examples, and there are lots of different models for focal adhesions. But these 3, I think, capture what we talked about today. So number 1 here is this one is how does a focal adhesion distinguish between soft and stiff ECM? So in simplistic terms, if you look at this cartoon, the rectangle here is the focal adhesion. This squiggly line is the ECM, and let's say that bond is simply a spring. And if you apply a force, what will happen on a very basic level is that this soft ECM will get stretched. As a result, the distance between the other parts of the ECM and the focal adhesion will become higher. So now this new adhesion that is waiting to attach to this black dot, the distance between the two has gone up because the ECM is soft. Now with the increasing distance, the probability of this bond formation will go down just based on proximity. Because of the soft ECM, the distance between the two has gone up. So the likelihood of new bond formation will go down. On the other hand, if it's a stiff ECM, and if you apply the same force, because the stiff ECM is stiff, it won't move that much. The distance between the bond on the ECM side and the cell membrane does not really change that much. So the probability of new bond formation does not go down. It remains high, right? So that is the basic idea, proximity idea. On a soft ECM, the proximity between the cell membrane and the ECM is not that good. On stiff ECM, it doesn't go down. So the probability of new bond formation is not hampered because of the ECM properties. So these 2 things are captured into this equation. This is the binding rate. Ru is the unbinding rate. So Z square being an exponential that is done to fit the data. And it is basically saying is as you increase Z, the probability of binding will go down. And if you just do a simple force balance, K is the spring constant of this bond, and you plug into this equation, the same equation, you can get a force relationship. Binding rate related to force. Higher force will lead to higher binding. The higher spring constant will lead to higher binding because this is the denominator and the negative sign here. And you can look at the how this one equation leads to a dependence of focal adhesion size with respect to ECM stiffness in this paper. But this is the basic concept. Another idea was about that actin filament velocity thing that I was talking about earlier. This number 2. So let's say ECM stiffness is here. It's a springs and series model, basically. If the velocity of actin filament is very high, and given in this equation, then the force developed is going to be low. And if you examine this equation, if the force is 0, then the velocity is highest. If the force is equal to the stall force, then the velocity must be very low. It will be 0. And that leads to this time relationship as opposed to force. And if you plot it for different stiffnesses, as you can see, higher the stiffness, the force development is faster. So the x-axis is time, y-axis is the force development. And for higher spring constant, force develops very, very quickly within 5 seconds and plateaus. On a spring, a softer spring, force doesn't develop as fast. All right. The third model is looking at thermodynamic equilibrium. This is a little more complicated. We are just comparing energies here. So on the left-hand side, this is low affinity integrins, meaning unbound integrin. On the right-hand side, psi h. This is the high concentration of high affinity integrins, and this is the force applied. So what is happening here is that if force is increased, I'll just focus on that last part. If the force is increased, then the energy level goes down, and the only way to compensate is by increasing the concentration of high affinity integrins. So this balance between psi h and this energy loss due to exertion of forces and the bond stretch is how focal adhesion keeps turning over, keeps growing. So if the forces and the bond stretch are low, there will not be a loss in energy. There is no reason for the adhesion size to go up, which is psi h. So rise in forces leads to adhesion clustering. And if the forces are reduced, the adhesion remains stable, it doesn't grow. All right. And this is the feedback loop I've been talking about. I won't talk about it more, but you can look at how it is implemented in terms of calcium ion transport in this paper, if you're interested. And I think I will stop there, and I'll give these slides. You can look over in 3D. Things are different. I only have a couple of minutes, I think. And I was going to talk about cell-cell versus cell ECM adhesions, but I won't today. Basic idea being, let me just go to the last slide. Just a couple of more minutes here. So let's say if it's a group population, so cell type A attaches to itself with a given strength. Cell type B attaches to itself with a given strength. Their intercellular adhesion is different. Cell type B attaches to the ECM in one way. Cell type A attaches to the ECM in a different way. Let's think about a scenario here. So let's say the ECM adhesions are quite weak in this ranking. Just look at this ranking, highest to lowest. The cell type A adheres to itself really strongly, and cell type B adheres to itself not so strongly. This is the predicted outcome. None of them wants to stick to the ECM. The dark ones really want to stick to each other, and the next in the ranking is the hollow ones. So this is the potential outcome of tissue formation in a way. And the next other ranking is, you can play different scenarios. This could be your homework. Think about it. Now, the highest probability, highest adhesion strength is of cell type A with the ECM, and then of B. And the lowest probable is cells don't want to really stick to each other. Then the outcome is going to be this. Because the darker cells, these ones want to stick to each other a little more than the hollow ones, and they all want to really stick to the ECM. So then the predicted outcome is this: you get a monolayer with sorted cells with each other. Other intermediate examples might be this: one cell type really wants to stick to each other, but not so much to the ECM. And the other cell type doesn't want to stick to each other at all, just wants to stick to the ECM. Then you might get this. Another example possibility is that the darker cells really want to stick to the ECM, but the hollow ones, not at all. Then you might be a dual layer of different cell types. So all this is saying is that by playing just the adhesive properties of cells, different cells with each other and with the ECM, you will get different types of tissue formation and cell colony formations that you see all over our body. So this is cell type dependent adhesion is what we are made of. And just by looking at these 4 or 5 different variations, you can come up with different scenarios for different disease processes or organ development and so on. And I will stop there because this gives you a lot to think about, this last slide, in terms of how adhesions are important in different processes of like disease and development. Right, questions?
This is
Oh, sorry, you go ahead.
This is Ethan from WashU. So on this last slide, it's just different for every cell, like the relative affinities.
Yes, exactly. So we are saying is that there can be different rankings for how cells are attaching with each other versus how they are attaching to the ECM versus how they are attaching to themselves. And if those adhesive properties have different rankings, they will lead to different outcomes.
Thanks.
You're welcome.
This is Chris from UCLA. Have you looked into whether myofibroblasts have a different adherence or type of cell adhesion versus regular fibroblasts? And in the context of like fibrosis?
I haven't, but I think there are papers showing that, yes. And myofibrils versus fibroblasts literature, I haven't followed that much, but in cancer progression, there are like metastatic cells versus normal cells. They have been shown to have different adhesive properties. And the way kind of it is measured is that these assays where you put a bunch of different cell types on a given substrate and just apply a centrifugal force, and whichever ones fly off, they are less adhesive. Whichever ones don't fly off are more adhesive to the ECM. And to look at the adhesion of cells with each other, there are these experiments where you would put one cell type onto the ECM and put another cell type onto like an AFM probe and lower the probe and make the cell-cell adhesion bond and then pull on the top cell. And that will give you a measurement of the adhesive force or the rupture force between two cells. They can be the same cell type or different cell type. So these are two different assays to kind of quantify everything that is on this slide. All of these don't need to be like hypothetical. They can all be measured. Adhesion between the cells and adhesions with the ECM of different cell types.
Thank you.
You're welcome.
Looks like we might
Okay,
be done.
so
Amit. Yeah.
yep.
Okay.
Go ahead, Ram.
No, that I just wanted to thank you, Amit, for the wonderful lecture. Does anyone have any last burning questions? Otherwise, we'll wrap up here. Okay, thanks again, Amit.
And we'll see you all back at your group links this afternoon for workshops.